Hello students. To determine the expected number of progeny with the genotype capital A, capital D, small r and capital Q, we need to consider the independent assortment of the genes capital A, D and capital R, Q. Given that the genes A and D are 26 centimorgan apart, so we can assume they undergo a recombination frequency of 26%. This means that the chance of a crossover between A and D during meiosis is 26%. Similarly, the genes capital R and capital Q are 12 centimorgan apart, indicating a recombination frequency of 12%. The test cross involves crossing the following genotypes capital A, small d, small r, capital Q crossed with small a, small d, small r, small q and small a, capital D, capital R, small q crossed with small a, small d, small r, small q. So here let's break down the expected number of progeny with the desired genotype. So first one capital A, capital D, small r, capital Q. This genotype can be produced through two different combinations. One is capital A, small d, small r, capital Q from the first parent and small a, capital D, capital R, small q from the second parent. Since these are independent events, we multiply the probabilities. Probability of capital A, small d, small r, capital Q is probability of A into probability of DRQ. So that will be 0.5 into 0.5 equal to 0.25. Now probability of small a, capital D, capital R, small q equal to probability of small a into probability of capital R, D, capital R, small q. That is also 0.5 into 0.5 equal to 0.25. Now the expected number of progeny with capital A, capital D, small r, capital Q genotype will be equal to total progeny into total probability of capital A, small d, small r, capital Q into probability of small a, capital D, capital R, small q. That is equal to 1000 into 0.25 into 0.25. So that is the expected number. That will be equal to 62.5. However, since we cannot have fractional progeny, we round to the nearest number, that will be 62. Second is small a, small d, small r, small q. This genotype can also be produced through two different combinations, small a, small d, small r, small q from one parent and again small a, small d, small r, small q from the second parent. So probability of getting small a, small d, small r, small q equal to probability of getting small a into probability of getting small drq. That is 0.5 into 0.5 equal to 0.25. Now the expected number of progeny with small ADRQ, that will be equal to total progeny, that is 1000 into probability of ADRQ, that is 0.25 equal to 250. Therefore the expected number of progeny with capital A, capital D, small r, capital Q is 62. Expected number of progeny with small a, small d, small r, small q is 250. Comparing the options given here, we can see that closest value of 62 is here 57. So which corresponds to option A. Therefore, the correct answer is said to be 57. I hope this video is useful for you. Thank you.